Hello and welcome to the next episode of Porous Media Tea Time Talks. We are part of the Interpor Academy and we welcome you here today. In the studio, as part of the team today, we have Kamalijt Singh from Harriet University, Javier Santos from University of Texas, and Nara Brandão from Federal University of Oberlin. Now we can go and introduce the first speaker. Our first speaker today is Song Lee. Song is a postdoctoral researcher in Professor Peter Kang's group in the Department of Earth and Environmental Sciences at the University of Minnesota. Song received his PhD from Seoul National University in 2015 to 2017 in chemical and biological engineering. He worked as a postdoctoral researcher at the Korea Institute of Science and Technology, KIST, before joining the University of Minnesota in 2018. His research combines experimental, mainly microfluidics, and numerical simulation to advance our fundamental understanding of interplay between biological, physical, and chemical processes taking place inside the porous and fracture media. Thank you, Song, for joining us today, and the floor is yours.
Thank you, Mohammad, for the kind introduction. And hello, everyone. My name is Sang Ni, and the topic of my presentation today is about how pore-scale flow can induce aggregation of microbes in porous media. This work was done in collaboration with Eleonora Secchi in ETH Zurich and under the supervision of Professor Peter Kang at the University of Minnesota. Before I move on, can everyone see my cursor? I have to go back and check. Yes? Okay, thank you. So let me begin by explaining what bioaggregate is and why it is important. So bioaggregate is a collective mass of microbes, organics, and inorganics, and it is commonly found in many natural and engineering systems. This fluorescence image is an example of multi-species bioaggregate generated in our lab. It is composed of MX torquons, shown in red, embedded in the aggregate of E. coli cells, shown in green. So these aggregates play vital roles in many natural and engineering systems. For example, biogeochemistry underground is reported to be mediated by units of soil aggregates, and bioremediation of wastewater is performed by activated sludge flux in a bioreactor. But interestingly, it has been reported that the clogging in porous media is mainly caused by these aggregates blocking the pore neck or pore throat regions of the porous media, not by the uniform growth of biofilm on the surface. However, up to date, we only have a limited understanding on how these aggregates are formed in porous media flow systems. And in our preliminary experiments with microfluidics with E. coli cells, we observed there was a preferential clogging at the pore throat regions of the chip. And considering there were a significant amount of researches that reported that pore-scale flow can impact attachment and accumulation of biomass, we hypothesized that there is a unique flow structure at pore throat that causes such aggregation of cells. So to test this hypothesis, we prepared a PDMS microfluidics chips with a single straight channel having a sinusoidal pore throat feature in the middle. And then we injected fluorescently active E. coli cells using the syringe pump into this channel and observed the cells in the channel using either fluorescent microscope or confocal laser scanning microscopes. During the entire experiment, the flow was kept laminar and the numerical simulations of flow was performed in COMSOL multiphysics. Here is the video from the result of the flow experiment on straight channel and the channel with pore through feature. I'll play the video on the straight channel first. Note that the video is fast forwarded and the real time in hours, minutes, and seconds is displayed on the top right corner. We see slow attachment and growth of the equalized cells in the channel, but no formation of aggregates in the span of 16 hours. And in case of channel with pore through it, immediately we see a rapid attachment and aggregation of cells, and once they gain enough mass, they were flushed downstream by the flow until they finally detach around 2.5 hours. I'll play the video again. It was interesting to us that this whole process of bioaggregation and the detachment happened so quickly within 2.5 hours compared to the straight channel case. So to analyze in more detail what we just saw, I prepared this plot. This plot shows the accumulation or the amount of equalized cells in the channel approximated by summing up the fluorescence intensity in the channel with respect to the time. The circles represent the result from pore through it channel, and the triangles from the straight channel. The color fills in the symbols indicates the data points corresponding to the image on the right. So if you look at the pore through it case, we see immediately upon introduction of the flow, the equalized cells attach at the pore through it, and then they accumulate, forming aggregates in the expansion side of the pore through it. And then once they gain enough mass, they slowly starts to detach from the throat, and then they show sudden disconnection from the pore throat, leading to this sudden drop in the fluorescent intensity at the later hours. At the same time, there was a negligible amount of accumulation of biomass in the straight channel. And upon closer inspection of our data, we define there are 3 stages involved in this bioaggregation process. They are attachment, shear-induced cell displacement, and cell accumulation. In the first stage, the cells are preferentially attached at the pore through it through the combinations of these 3 flow structures that arise from the constriction and expansion feature of the pore through it. In the second stage, the attached cells were pushed downstream by shear. I'll show you this video clip again to clarify what I'm saying. So if you see this video, you see as the cells attach at the throat, they were at the same time pushed downstream in the region of expansion. And then what's interesting is that while this is happening, the pore throat remains relatively a fairly constant pore opening. So we suspected that as the cells attach, clogs the pore, narrowing down the cross-sectional area. It increases the flow velocity because we imposed a constant flow rate boundary condition, which increases the shear stress. And once we suspected that once the cells cross this boundary and attach within in the inner side of the channel, beyond this boundary, they're exposed to a shear stress that is critical, at which the cells are mobilized, which causes the cells to be pushed downstream. In the third stage, this cycle of attachment and shear-induced cell displacement repeats until the enough cells accumulate in the region of expansion and form aggregates. And in the remainder of this presentation, I'll mainly talk about the second stage, but feel free to ask me any questions afterwards if you have questions in the other stages as well. So from previous slide, I suggested that there exists a critical boundary at which critical shear stress happens. And at this, above this critical boundary, when cells attach, they are exposed to critical shear stress, which mobilizes the cells, pushes the downstream, where the shear stress drops back below the critical shear stress value where they are accumulated. So in order to estimate this critical shear stress value, we made two additional hypotheses. One is that this critical opening bounded by this red boundary will increase with respect to flow rate. That is quite obvious because if we increase the flow rate, the cross-sectional area required to reach the critical shear stress will increase, pushing this boundary outward. And the second assumption hypothesis is that this shear stress takes place along this line at different flow rate will have a similar value because they are all critical shear stress value. To test the first hypothesis, we performed the same experiment, but by changing flow rate from 0.1 to 0.3 microliter per minute. And this is the result. We put a cross line y prime across the pore through it, and then analyze the fluorescent intensity to estimate the critical opening width. So it shows that as we increase the flow rate, the critical opening width increases with respect to the flow rate, which confirms that our first hypothesis was true. So in order to move on to the next hypothesis, we estimated the critical the shear stress at this boundary. We did that in console multiphysics by incorporating the biomass as impermeable and no-slip boundary cylindrical surface. And the degree of intrusion of the biomass was determined from the experimental data in the previous slide. So here is the result. The shear stress was observed at the peak or the narrowest point of the throat, and they are designated as stars. For the surface, biomass covered surface, and black circles for clean throat surface. And surprisingly, at the different flow rates we tested, the maximum shear stress value converged to a single value of 1.8 Pascal, which we suspect that this is the critical shear stress value. And then we back calculated by using this plot, we back calculated the flow rate at the clean pore throat, which yields this critical shear stress value, which was the 0.75 microliter per minute. And we performed the microfluidic experiment around this flow rate to see how this critical shear stress affects the bioaggregation process. So here's the result. The top half of the image shows the experimental result from the fluorescence microscopy, and the bottom half is the result from the numerical simulation showing the shear stress field. The blue region shows the shear stress was below the critical value, which was 1.8 Pascal, and the red area shows the region that was exposed to critical shear stress or higher. And as you can see, when the flow was below the 0.75 microliter per minute, the aggregate showed chunky biomass morphology, while at or above the critical shear stress, the bioaggregates show streamer-like morphology. We suspect that this is because that as soon as the cells attach at the porthoid, they are immediately mobilized downstream, forming this stretched biomass shape or streamers. And if we increase the flow rate further, where almost everywhere there is the entire region is experiencing the critical shear stress, no detectable attachment or aggregation was observed. Then we related this concept of critical shear stress to the concept of critical pressure. We run the same experiment, but imposed a constant pressure boundary condition, which was at 31 millibar, which yielded a flow rate below the critical shear stress value. And upon the introduction of the equalized cells to the channel, we see immediate clogging of the porthoid and reduction of the flow rate due to the clogging. And if we introduce the flow at higher pressure, which yielded a flow rate that yielded a shear stress higher than the critical value, the porthoid remained open. So in conclusion, we found that there exists a critical shear stress above at which aggregates take streamer-like morphology, and below at which they form chunky biomass morphology. And this concept of critical shear stress was related to the concept of critical pressure in clogging. For example, above, this could suggest that above there is a critical pressure that needs to be maintained for a porous media containing porthoid in order to keep the porthoid remain open. And we found that such bioaggregation process can significantly reduce the amount of flow through the porthoid, which can subsequently have impacts on transport and reaction phenomena in porous media. And here are the funding sources of my research, and thank you all for your attention.
Thank you very much, Sang-Li, for this great presentation. It was very insightful because you show a very interesting phenomenon, and for me it was kind of counterintuitive to see that the cells first start nucleating where the velocity is the highest, right, which is at the pore throat. So
right.
I wanted to ask you, you mentioned that that's due to the collision of the streamlines, right? That they all go through the pore throat, and that's where the cells start nucleating. Do you think this is due to the higher concentration that's in that region, or how do you explain that phenomenon in layman words?
I would say yes. But more specifically, let me show you this slide. So if this phenomena is also called flow line interception, and is actively used in capturing air particulate in some filtration processes. So what happens in this scenario is that when a fluid containing particles are introduced into a constriction where the streamlines are densified, the streamlines near the surface, they become dense, and the particles that were traveling on those streamlines become concentrated or move closer to the surface, which increases the chance of collision onto the surface.
Oh, excellent. Thank you. And my last question is, do you think that surface chemistry of the solid has something to do with this, or is it mainly the pore morphology?
Well, I would say it will have an impact on especially the attachment stage because the strength of the attachment and the probability of complete attachment to the surface will determine by the interaction between the cell surface and the surface of the walls. But once the cells form some kind of layer on the surface of the pore fluid, then it's the interaction between the cell and cell. So I think at that point that chemistry will have a less effect.
David,
Sounds all right.
I have a quick follow-up question, if you allow me, Javier.
Yes, please.
Okay. I think it's along the first question what David asked. Do you have any critical radius of pore throat that will help this? Let's say because you looked into flow rates, and then you looked into the what's the diameter or the radius if when these microbes are they forming things. But let's say if you vary the size of the pore throats, do you have information, or maybe you've done some experiments where you would say the pore throat doesn't affect if your pore sizes are large, throats are larger, or
Yes.
maybe it depends on aspect ratio. I'm not sure, but just considering this.
Well, I need more detailed set of experiments to confirm that, but this is the preliminary experiments we performed by varying the degree of pore throat. And we see as at the low ratio of the pore throat size to the channel opening, the channel size, we see increasing as the degree of pore throat opening gets smaller. The accumulation or attachment of the biomass is increased. So we, as you suggested, we think it should be some kind of critical value that that causes the rapid increase in the attachment of the cells and accumulation of the cells. But we have not defined at what value it causes it.
Okay. So just again, it's just a follow-up, quick question. It's a naive question. Let's say the first image on the left, on top left, the throat size, because we know the biofilms are forming. If you take the whole tube with the uniform diameter of that size, would you expect to form biofilms again? That means no pore throat, only single capillary of that size.
I would say no, because the idea of this preferential attachment of pore throat is the constriction feature that densifies the streamlines near the surface. So, but if the whole channel is made of a single straight channel but with smaller diameter, the density of the streamlines will remain constant along the length of the channel. So I would say no.
Okay.
And so also we have some questions from the audience. So I'm going to quickly go through them. Marcel here is asking that if you know what's the nature of the force that makes the cells stick to the chip.
The attachment is driven by the interaction between the cell surface and the surface of the wall, which is the chemical and physical interaction. So I would say, depending on the material, it can be electrostatic or van der Waals, but yes.
Excellent, thank you. And our last question from the audience is from Susmit. He's asking which will be more realistic in a modeling setting. Do you think we should model permeability reduction as a function of the reduction of the porosity that you're seeing in your models, or by attached biomass accumulation? Like, which modeling approach would you suggest for people that are doing bigger scales? He's specifically asking for sandstone, so I guess we're thinking about the more ideal situation.
So if I understood the question right, the question was whether we model the biomass increase or the reduction in flow rate?
Yeah.
My training is not a modeler, so it's difficult for me to give insights on that. But my intuition is that biomass is very complex, and it can have the time frame that I monitored is quite short period of time compared to the physiology or metabolism that happens in the biomass. For example, the change in gene expression and the growth is, I believe, it's quite a little bit longer than the time scale that I'm observing. So modeling the biomass could be challenging, I think, but the modeling the flow rate will be a lot simpler approach, I think.
Excellent, thank you. Yeah, I guess the conclusion is it's a very complex process that we need to still investigate. Thank you very much, Sang-Li. So now, without further ado, I'm gonna introduce our next speaker. Our next speaker is Mahdi Mansouri. He is a PhD candidate at the Institut des Sciences de la Terre in France. He has a background in petroleum engineering. He's been modeling interfacial phenomena for quite a bit. And now he's going to show us his current research on CO2 storage in saline aquifers and the pore invasion dynamics that happen in this. So, Mahdi, please, the floor is yours.
Yeah, thank you very much, Javier, for a very kind introduction. And I'm very grateful that I'm invited to give a talk here in Porous Media Teton Talk series. Today I'm going to talk about pore invasion dynamics and transition from viscous to capillary flow regime during drainage in porous media. First, I will introduce you to the subject as a context of CO2 sequestration. Then I will present the research aims, and then I will show the approach that I took. And at the end, I will present the results on the fronting stability and the link between the Haynes jumps and corner flows. And finally, I will conclude through the work. CO2 sequestration has been known to be one of the most promising mitigation methods of CO2. CO2 is being captured from the atmosphere or power plant and then being injected to the aquifer subsurface. But there are still some main challenges that remain unanswered. And what is the exact storage capacity? What is the storage mechanisms in CO2 sequestration? And what are the risks associated with CO2 sequestration? To understand these phenomena and answer these questions, we have to deep look where this displacement is happening, which is very beautifully depicted by the Stifel et al. We can see CO2 is displaced in the aqueous phase, and with different fluid configuration, CO2 can displace the aqueous phase through each of these capillaries. At each flow path that CO2 takes, CO2 will be in contact with the water and can dissolve into the water. And this alters the thermodynamic equilibrium that exists in the porous media. And this thermodynamic alteration will trigger a chain of reaction in the porous media, which might end up with precipitation or dissolution of the sulfates, and petrophysical properties will change, which in turn changes the flow properties. So we're dealing with a highly dynamic system, and both chemistry and the physics of the flow are important. But now we just only focus on the physics of the transport. Lenormand et al. Were the first one who kind of categorized different fronting stabilities based on two dimensionless numbers. Mobility ratio, which is the ratio of the viscosity of the displacing phase to the viscosity of the displaced phase, and the capillary number, which is the ratio of the viscous forces to the capillary forces. They distinguish three different flow behavior: the stable front displacement, capillary flow regime, and viscous flow regime. Lenormand investigated different pore invasion mechanism at different flow conditions. Haynes was the first one who understood this movement of the interface, apparent smooth movement of the interface at some conditions is not valid, and interface is jumping through the porous medium with a rapid movement. Singh et al. Related the frequency of these Haynes jumps to the ramification of the front, and Armstrong and Mobius et al. Observed the non-locality of the capillarity effect in the porous media. Roman et al. And Blue et al. Reported the disturbance of the wetting fluid far from the interface while the invasion was happening. In all the previously mentioned works, the link between the pore invasion mechanisms and the front instability is still missing. And all the previous works, the porous media is very complicated and pores are very well connected, that if we observe any instability, it's very difficult to relate this instability to a specific phenomena happening in the porous medium. So to overcome this problem, we take the pore doublet model, which this concept was first presented to explain how the petroleum was being trapped during the water flood in the porous media. By modification to this model and adding some pores and throat to each of these capillaries, we will have a succession of pores and throat as our porous medium. We have a high resolution camera coupled with a microscope, and the flow is being regulated with a pressure regulator in the porous medium, and then we are able to monitor the displacement in our microfluidic experiments in a nanomicro lab, which is a part of European research infrastructure here now in Orlando. We use PDMS microchips, and the wettability is altered toward the fact that every time we are at the drainage condition, we use different fluid pairs to have different mobility ratios, and we go from very low flow rates to high flow rates to have different flow regimes. Here at viscous flow regime, high flow rates, you can see the non-wetting phase invading through the porous medium. Meniscus position for different mobility ratio is depicted versus time, and time and interface position are respectively normalized for the time that meniscus takes to invade one capillary and the length of the capillary. We can see meniscus is able to invade both of the capillaries simultaneously, and the viscous forces are dominant here at this flow regime. And also what we can observe is that the transport of the meniscus in both of the capillaries are very similar. But if we look at a single pore scale, we can see at this condition, non-wetting phase has not been able to invade the whole cross-section of the pore. And there has been a layer of the wetting fluid left behind at the corners of the porous medium, which kind of implies that maybe at this condition, the viscous coupling between non-wetting and the wetting fluid must be considered while we are upscaling from pore scale to higher scales. When we move at the capillary flow regime, very low capillary numbers, we can see that the invasion has completely changed. Meniscus is able to only invade one of the capillaries. Capillary forces are dominant here, and Haynes jumps becomes the most important invasion mechanism. Meniscus pins at the throat and then suddenly jumps to the next pore, and then there is a waiting time when the meniscus is pinning at the throat. At a single pore, what we observe is it's very different from viscous flow regime, and here the non-wetting phase has been able to invade the whole pore. But what is the nature of this waiting time while the meniscus is pinning at the throat? To answer this question, we seeded our wetting phase with some particles to see whether there is any dynamic movement while the meniscus is waiting at the constriction. And if we subtract the particles from the background, we can see definitely there is a flow in the body of the wetting fluid. And if we perform particle image velocimetry, and if we average the velocity vectors here at the pore, we can see there is a continuous flow coming from the corners of the fluid to the body of the wetting fluid. But how does this happen here? Let's consider a meniscus pinning at the throat, and it wants to invade the next part, which is more constricted and tighter. So it implies that the capillary pressure would be higher at the next step. And considering the wetting pressure, non-wetting pressure constant, It means that if the invasion wants to happen, the wetting pressure at the tip of the meniscus should reduce. And considering at the corners nothing has changed, so the capillary pressure would remain constant, which implies that the wetting pressure at the corners will be higher than the wetting pressure at the meniscus if the invasion wants to happen, which provides a pressure gradient through the corners and probably a flow from the corners to the body of the wetting fluid. Here you can see my very first experiment in the lab here. Back then, I have a single capillary, and I'm withdrawing the fluid from one side, and the non-wetting phase is invading from here. Back then, I expected to see the meniscus coming to the channel and invade continuously the porous medium. But what I was observing is that the meniscus slowed down, and there is a continuous flow in the body of the wetting fluid. Now I understand by increasing the capillary pressure while the meniscus is invading the tighter parts, the capillary pressure increases, and this increase in the capillary pressure makes the corners of the geometry to drain to the body of the wetting fluid. To have an insight about the dynamics of this phenomena, we use foam drainage equation, which relates the velocity through the corner to the surface area and the modified viscosity due to the shape of the corner and the pressure gradient. And the capillary pressure also could be estimated from the changes in the contact line between the non-wetting phase and the solid phase, the non-wetting phase and the wetting phase and the contact angle, and the changes in the surface area of the non-wetting phase. If the model is single phase, considering that the meniscus is pinning at the throat and the pressure at here will be the tertial pressure for the invasion of this throat. At the time zero, the wetting phase has wetted all the corners, all the surface area of the channel. And at the end, due to the literature, the radius of the corner of the wetting phase at the corners will be 1.2 times of the width of the capillary. Then we will have the velocity through the corners versus time. We can see at the beginning, we have an initial velocity which decreases by time, and until we have basically no flow. And by increasing the viscosity, we reduce the initial velocity, but the flow will be more continuous. We can perform the same sensitivity analysis here for the contact angle. For the full wetting, we have the highest initial velocity, but the corners will be drained faster. And by increasing the contact angle, the initial velocity decreases, but the flow will be more continuous. If we perform a PIV at this throat while the meniscus is pinning here in our experiments, and comparing the model with the experiments, we can see a very good compatibility for the fluid behavior and the fluid velocity in our experiments as well. It starts with a high velocity, and then it decreases until it reaches a certain value. So to conclude all the presented work, we were able to highlight relation between the micro-scale behavior, micro-scale front behavior, and the front shape. So different pore invasion mechanisms were identified at different flow regimes. We observed also maybe the residual saturation can be different from each flow regime to the other. And also this idea of leaving some fluid behind at viscous flow regime might change the chemistry which is going behind at the CO2 sequestration state. We observed that Haynes-Jones is clearly the most important drainage mechanism at low capillary numbers. And we observed that there is a dynamic of the corner wetting fluid that is left behind when the pore is being invaded at very low capillary numbers with Haynes-Jones. And thank you very much for listening to my presentation.
Hi, Majid. Thank you very much for your presentation. I really appreciate it. I like this research topic.
Thank you very much.
First, I'd like to invite the audience to type your questions in the text box. Please feel free to type your questions. I guess we've got one here that I can share with you. Okay, so it's from Arjen. Thank you for your presentation. Do you have an idea why under capillary dominant condition, the non-wetting phase only flows in one of the two flow pipes?
Okay, thank you very much for the question. In fact, when we have capillary dominated flow regime, the thing is the capillary forces are very dominant here, and they start to, like, the flow is very low at it can only invade one of these capillaries, and while maybe there is a small deflection between these capillaries that that can change whether the meniscus can invade the pores or not. And this is and while the meniscus reaches the pore body, then the capillary pressure reduces here and completely it can completely damp the invasion in the other capillary as well.
Okay.
I have a quick question. I thought this what you showed was very interesting, because it seems that to understand hinge jumps better, we need to understand the dynamics of the wetting fluid as well, not only the non-wetting, right?
Yes, exactly.
Yeah. And meanwhile, we also got another one. It's from Marcel, and very interesting talk, Maja. Thanks a lot. Do you think the thin wetting phase film remains connected to the bulk of the wetting phase during the whole experiment? I'm also curious about that.
Yeah, okay. Very, very great question, in fact, here. What we observed during the experiments, definitely there will be a film flow also inside while we have these corneal flows as well. There will be a film, and also this film is most probably being drained while the corneals are being drained. I can see that kind of in my experiments in the image footage that I have, but maybe it is not measurable here. But definitely there will be a film flow as well while we have the corneal flows.
Okay. Okay.
Nara, I have a question if you have time left. Thank you, Mahdi, for this fantastic talk. It's a more technical question. Maybe it's you changed the wettability of the system with the PDMS. Can you mention how you changed it, with the chemically or something else, or by changing?
Okay, yeah, because we chose different fluid pairs here.
Okay.
We have different methods to change the PDMS toward completely wetting or completely non-wetting, because when we were using sometimes silicone oil as the wetting phase, so we use some saline to cover the capillaries. We injected some saline vapor into the capillaries to turn them completely non-wet or water repellent. And when we had the water as our wetting fluid, we used either the plasma chamber to plasma treatment to change the wettability, or we injected some PVA inside the channels to deposit on the surface and makes it water wet.
Okay, so I think you didn't use any gas in there, because I would imagine that PDMS is permeable to gases, and
yes,
it might have some difficulties there, right?
Yes, definitely. In fact, I have a set of experiments with air and water, but air wouldn't be a problem because the atmosphere also is filled with the air, so there would not be any transport in that.
Okay, thank you.
Anytime.
Great, and I just want to ask you a quick question from my side. Have you evaluated the influence of pore space heterogeneity in your model?
So for now, we just wanted to have a completely homogeneous porous media to have the idea of these identical daughter channels as a pore doublet model. But definitely the effect of heterogeneity could be very interesting, especially while the front has passed and some of the pores has not been invaded. Because especially at low capillary numbers, at capillary flow regime, what we can observe is that while the invasion is happening, the interface at other pores show a response due to the Haynes jumps. So there will be some oscillation at the interface while the Haynes jump is happening. So definitely that could be interesting.
Okay, yes. Thank you. And we got another one from Cyprus. Thank you for your talk. If I understand correctly, you used air water for your blue curve experiment. And so do you think that air compressibility has an impact on the jump dynamic?
Okay, yes. This is a challenge for the system of air and water, like the compressibility of the gas system. But in fact, I would say maybe it has a slight effect on the interface movement, because we can see the same trend in all the other experiments. But I also would say, like, because the gas expansion is only for one single pore, maybe the effect of the gas is ignorable here at very low capillary numbers. But definitely, if we can choose a set of incompressible fluid pairs to test this, that would be great.
Okay, thank you. Yes, great. So thank you for the participation of the audience and also from my friends here in the studio. And once again, thank you, Majid, for your great talk.
Yeah, thank you very much to invite me to give a talk. Yeah.
You are welcome. So I guess we can, we would like to invite you for our next session. And it will be on 2nd of November at 10:30 CET. And we are going to have 2 other great talks from Ionis, and who will talk about the reservoir on a chip toward the development of 3D printed rock alike micromodels. And the other talk will be from Javier, and who will talk about the micro-scale biophysical drivers from microbial colonization in porous environments. And so once again, thank you for your participation. We appreciate. And on behalf of our team, I'd like to invite you for our next session and see you there. Bye bye. Thank you.